So Vanya asked me to come today, and she said, well, can you come give a talk? And I said, but Brett knows everything about prime flow, and Valentina knows everything about the actual research, which you're going to hear presented both after me. So while they know more about me, about all those other things, I am a big flow cytometry geek, as Vanya politely just introduced. So what I'm going to talk about really is the field of flow cytometries for the State of the Union, what technology exists, why we want to look at RNA species and protein species, and the different types of platforms that are out there to do that. So we'll start by talking about what flow is at a very high level, because I can't otherwise do that in 20 minutes. So flow cytometry is very useful because you get to look at individual cells one at a time, like Mike had introduced. Averages can be very misleading. We want to see all of the different individual cells as individual data points. And we can analyze them very, very quickly, and we can do it in a highly multiplexed manner. So we get our fluorescence. I don't like doing microscopy and drawing regions of interest, and then 2 hours later you get data and all this information, and then you realize the segmentation wasn't good. Go back and do it all over again. Flow cytometers give you your fluorescence information in real time immediately as you put it through the flow cytometer. You can do, let's say, 30 colors at a time, very much independent on the instrument, and you get high throughput of cells, 10,000 cells per second. So you can look at rare subsets, you can look at circulating tumor cells, you can look at really finite immune cell populations that are regulating your disease, etc. However, we lose a lot of information that you typically retain through microscopy. So you don't get morphological information, you can't see nuclear and cytoplasmic, they're not adherent on a plate, you can't see what things look like. You have to dissociate a tissue, you have to take it off of a plate, or you can take cells like blood cells that are normally in suspension and just put them straight into the instrument. Once you've done that, and sorry, I shamelessly show pictures to my children on my talks, you'll have to just suffer me that, you can do a variety of applications. This is a bit of a laundry list of things that you can do, but you can look at DNA content, cell proliferation, viability, you can look at different cell protein amounts, as well as cell proteins that identify what type of cells those are. There's a million things that you can do, and then you can also use those things to sort out the cells, isolate them, co-culture them, ask genetic information about those cells, etc. The point I want you to take away here is essentially if you've got a molecular probe, fluorescent probe for something, including prime flow, you can ask those questions about a cell, and you can do 20 or 30 of those things in a tube at a time. So you can save precious clinical samples. Now, not only can we do this at a cellular level, but as I'm sure many of you are aware, there's now a lot of interest in things like exosomes and extracellular vesicles. So these are submicron pieces of cells that do a lot of different things that we certainly don't have time to get into, but they carry microRNAs, link RNAs, messenger RNAs, secondary messengers, oncoproteins, et cetera, et cetera, et cetera. They can take that from your tumor, they can go over to neighboring non-transformed cells and elicit changes there that are helpful for the tumor or helpful for a therapeutic intervention. The point being here that we can not only look at protein, but also RNA. We can not only look at cells, but subcellular things like extracellular vesicles that are a very hot topic in the field to date. Now, why do we care about RNA, right? Protein's the doer, it does all of the things that we want to do. The problem with looking at just protein is that when we fix proteins, they change their epitopes. It's hard to detect them. Limit of detection for proteins can be in the dozens to hundreds, depending on what we're talking about. Limit of detection for RNA species is much more favorable overall. RNA changes much earlier than your protein changes. And as I'm sure you've all heard, we've got this antibody crisis, and we're blaming it for all of the non-reproducible data, not the summer students. But the point is that antibodies do lend to a lot of variation in science, and they do lend to a problem. And if you're looking only at protein, that does create a bit of an issue. So it can be very advantageous to say, look at CD3. CD3 is going to be fine. You're going to always find T cells on CD3. Whether or not you see certain other things or whether you have to move to a transcript is a totally different question. And there's not always antibodies available for the things that you want to look at. So if you want to look at one of those things, it's much easier to adapt something like a prime flow assay and look at those things. So we're going to take a step back for a second before we talk about the different types of flow cytometry that exist that allow us to look at things in pros and cons. And we're going to go at a really high level as to what flow cytometry is. So in flow, you either take naturally suspended cells like blood cells or suspension cultured cells, or you take associated tissues or tumors or liberated cells off of plates, and you introduce them into your flow cytometer, and they're all side by side, and they're kind of running around, and we narrow down the stream of delivery such to the point where we deliver individual cells in a single file line past lasers. Those lasers excite the fluorescent molecules that you're looking at it, be it a molecular probe, a branched DNA construct, an antibody, or whatever. That fluorescence information is then collected by a lens, just like you would on a microscope. For a few technological reasons, sometimes this can be a little bit more sensitive. And again, we get this information in real time. Because these cells are going past these lasers and being scanned very rapidly, we don't need to worry about things like photo bleaching. And we do collect that fluorescence information immediately. That fluorescent light then goes past various filters and mirrors, and the red light goes to the red detector, the green light goes to the green detector, so on and so forth. This is a little bit dependent on the platform, and then it's amplified by some type of detector, often a photomultiplier too. That makes it very, very sensitive, and again, rapid speed, like I've mentioned. Now, the question is, how do we do this? And this is where the devil is in the details. There's many different approaches from different types of cytometers to analyze these kinds of things. And a traditional flow cytometer, we've just got a diagram here where the blue line here is our excitation laser, blue laser, 48 laser, exciting FSE very, very well at 488 nanometers, and then it emits a spectrum of photons. So it doesn't just spit out green photons, it spits out a probability curve of a whole bunch of different photons. We collect in this 530/30 bandpass filter 30 nanometers worth of light spectrum. Okay, that is not all of our signal, but that's what we count as our signal for this FSE fluorophore. The problem is that FSE also emits in the orange spectrum in our neighboring detector here where we would pick up PE or microRNA. Okay, now in this very simplistic example, what we end up getting the cytometer to do is quantitate that part of our signal that we're going to count as our full FSE signal. It might only be about 40–50% of the signal. And then quantitate the amount of inappropriate spillover signal in neighboring detectors, and here we're only showing one of them. Then we're going to say this fake full signal that we have here, which is only a part signal, is X amount. This little orange piece of the pie is how much of the green piece of the pie proportionately. Okay, it's 13%. So now I'm going to take this fake full signal, I'm going to take 13% of it and subtract it from that neighbor channel, and we go back down to a baseline of 0. Okay? Now, the problem with this is that the pieces of green pie and orange pie depend on your instrument setup. So you can't mess around with your instrument once you have it set up. The other problem is this is a really, really simple version of a two-color assay. And this can be complicated and you can mess it up. You can imagine when you have 20 colors because you want to look at a whole bunch of different immune cell phenotypes contributing to your cancer or whatever disease you're interested in, the overlap there becomes incredibly, incredibly difficult. But this is the traditional approach. We've been doing it since the 60s. And other folks, really smart folks over at the U of T, came up with DVS Sciences, which is now Fluidigm, and they said, you know what, instead of using these really, really broad fluorescent peaks that we have, and you can see here that overlap once you get into multi-parametric testing, we're instead of using a fluorophore, sorry, I'm not using this one around here, but instead of using this fluorophore up here that has a broad spectrum of emission, we're going to use metal tags on our antibodies. And those metal tags get detected by the cytometer by a mass spectrometer, and they don't have spread. Atomic mass is atomic mass. It's finite. It doesn't go across different atomic masses overall. And as such, you can multiplex 30 or 40 antibodies at a time, which means that this is great. We can do more immune cell types, etc. However, not a lot of people make antibodies or probes associated with metal tags. There's limitations there as well. Most recently, what flow cytometry has tried to do to increase your ability to unmix this mess is instead of dealing with relative pieces of pie and really complicated linear algebra, there's spectral flow cytometry. So this is what the newest thing is, and what happens is instead of taking slices of the light, we take all of the signal in spectral flow cytometry. So the way that works is we take the entire spectral fingerprint for, say, FSE in this green line here. We take that whole signal, and that component curve has a very identifiable shape to it. Also, your unlabeled cell will have autofluorescence shown in red, and that also has a very characteristic shape to it. And then you multi-label all of your cells, 30 colors, 40 colors, whatever it is, and you look at the component overlapping curves and you unmix them just like you do in deconvolution microscopy. Now, the electronics are fast enough to do this at 10,000 cells per second on a spectral flow cytometer now, so we can do this. And this is highly advantageous because, one, you can multiplex more, so you can use all of these prime flow probes, but you can also use lots and lots more protein probes as well. So you get more information out of your sample. The second thing is when you're doing a lot of fixation, like you would for an RNA assay, you can get a great deal of autofluorescence depending on your sample, or if it's otherwise fixed. And that's going to contribute to this background measurement, and typically in a normal flow cytometry experiment, create a lot of noise that masks your signal. In spectral flow cytometry, you can actually identify that autofluorescent signal, you can subtract that signal from your sample, and you can increase its sensitivity. And also, instead of taking just a slice of the pie, you're taking the whole pie. So typically, more multiplexed, higher signal-to-noise ratio. And it also means that you can have things that are horribly overlapped that you would not otherwise be able to unmix. So this is a fluorescence emission for ExaFluor 610 and eFluor 610. And you can see in red the window that we would collect on a traditional flow cytometer is almost exactly the same. There is no way you are unmixing those things or compensating those things on a traditional flow cytometer. However, the ExaFluor has a characteristic change in the shape of the curve here, and the slope here and here are very, very different. So that shape difference means that a spectral flow cytometer can actually identify your T cells and your other lymphocytes and unmix those populations really, really well. It's good enough to the point where you can actually, although you never really would, but you can, if you're just a geek, want to unmix ExaFluor 88 and FSE, even though they are like 2 nanometers apart on their curves. So that's sort of the newest rendition of flow cytometry instrumentation that allows you to look at highly multiplexed assays. In addition, there are imaging flow cytometers. They tend to be a bit more of a niche application, but what they do is they take pictures in all of the different channels of your cells as they go through. So you retain morphological information, even though you still need to dissociate tissue. But allows you to look at some things like cell interactions, so immunological synapse formation, autophagy, cell cycle, micronuclei, things like that, which would have some degree of relevance to a cancer audience. But this is slower than typical flow cytometry, and this is all collected off of one camera, so you can't change the sensitivity of different detectors. If one guy's low and one guy's too high, you have to change the detector sensitivity across the board. So there are some limitations, but it's of interest. The last type of flow cytometry, which I think is the most interesting for a cancer audience, is imaging mass cytometry. So this uses the metal-based probes that we were talking about when we were looking at highly multiplexed 40-color assays. But instead of doing a suspension cell sample, what you do is, just like in histology, you keep your section tissue, you stain with this 30 or 40 color panel of different metal labeled antibodies on your tissue. You then come along with a laser and zap subcellular sized spots. So you do have subcellular resolution. I think it's down to 2 or 3 microns, but don't quote me on that, I might not be right. And then that information gets atomized and sent to a mass detector, and it reconstructs, based on the XY position of each laser spot, an image of your tissue. So now you can look at stroma, you can look at vasculature, you can look at invasiveness, you can look at all of these different things as you segment into cells, and you get that multiplexed information. You also get information on who plays with what neighbors, and if you play with these neighbors, you become more of an aggressive disease, and if you play with those neighbors, you become a less of aggressive disease. So it has a lot of morphological capacity, and it allows you to mix hypothesis-based research with discovery-based research as well. So those are the platforms that exist that are out there, and also autofluorescence doesn't play a role in any of the math things because they don't have anything to do with light. But everyone's got, at the end of the day, everyone has a different niche application, and everyone has a different way of their cytometer being more sensitive or higher throughput or whatever. And you need to determine which of those platforms is best for you to do an assay. So with that, I will wrap it up. And if you have any questions about flow cytometry, or if you want to do any of these assays, by all means, speak to me now, speak to me later, email me, my information is up